Okay, hi everyone. I'm Mohamad Zoabi. I'm a postdoc at Stanford University in Professor Guzani's lab, and I'm gonna share with you today the results from a published JBC paper talking about the lysine methyltransferase methyl-21C, a work that I did in my first year as a postdoc at Stanford. So first, just with some introduction. Methyl-21C, as I mentioned, is a lysine methyltransferase. As you can see here, the lysine methyltransferase, or KMT, catalyzes the addition of methyl group to the lysine amino acids, where S-adenosyl methionine, the SAM, is the donor for the methyl group. The lysine can be mono, di, or trimethylated by the methyltransferase, where lysine demethylases, or KDM, are responsible to erase the methyl group. The activity of lysine methyltransferases are very well studied regarding histone methylation, but also the field of protein or non-histone methylation is growing a lot in the last few years. So methyl-21C belongs to the methyl-21 family that includes methyl-21A, B, C, and D. Alignment of the methyl-21 members shows that the key residues that constitute the catalytic bucket are conserved. It was shown that methyl-21 family methylates non-histone proteins, where methyl-21A methylates HSP70 proteins, methyl-21B methylates EF1 alpha, and methyl-21D methylates VCP protein. Different from the other methyl-21 members that are expressed in several tissues, methyl-21C is a skeletal muscle-specific methyltransferase. Two exciting papers showed the importance of methyl-21C in muscle biology, where knocking down methyl-21C leads to muscle weakness and autophagic vacuolose accumulation. In addition, methyl-21C are reported to be implicated in cancer, but today I'm not gonna talk about the involvement of methyl-21C in cancer, maybe in the near future, but I will show you the new identified substrate of methyl-21C, ARS1, show you that methyl-21C methylates ARS1 in vitro and in cells, but does not methylate the other reported substrate, and that methyl-21C methylating ARS1 in a muscle tissue. So first, to investigate methyl-21C biochemical activity, we took advantage of the available crystal structure to generate catalytic mutant of methyl-21C by mutating two amino acids, tyrosine and tryptophan, which are responsible for the binding of the methyl donor, SAM, S-adenosyl methionine, that you can see here in red. To test the catalytic activity of the wild type and the mutant, we looked at the automethylation signal, a phenomenon we have observed for many lysine methyltransferases. Here, I used recombinant proteins and radioactive S-adenosyl methionine in vitro methylation assay. Results show that no methylation signal of the mutant compared to the wild type. We postulated that methyl-21C mutant could be used as a potential negative control in an unbiased experiment aimed to identify bona fide biochemical activity. So next, to identify the methyl-21C substrate or substrates, I used two-step fractionation protocol coupled to in vitro methylation assay and mass spec. Since methyl-21C is at cytoplasmic proteins, that cytoplasmic fraction further fractionated by size exclusion column using a FPLC machine. And every fraction was divided for two samples, which used for in vitro methylation assay with the wild type or the mutant recombinant methyl-21C that we generated. Here, we detected a strong methylation signal around a 100 kilodalton in three different fractions, indicating that we have a methyl-21C potential substrate, since this methylation signal appears in the presence of wild type, but not the mutant methyl-21C. So to detect the identity of the substrate in this fraction, I used mass spec analysis. And interestingly, we found that the methylated protein is alanine tRNA synthetase, AARS1, which is methylated at lysine 943. ARS1 plays a critical role in translating the genetic code via the high fidelity linkage of alanine to its cognate tRNA. To test if ARS1 is directly methylated by methyl-21C, I did in vitro methylation assay again with our recombinant protein, now not with the lysate, with our recombinant ARS1, and I detected very strong methylation signal of ARS1 in the presence of wild type, but not the mutant metyl21C. Furthermore, substitution of the methylated lysine K943 to alanine in the full-length ARS showed that there is no methylation. The mutant abrogated metyl21C methylation on ARS1. Based on this data, we concluded that ARS1 in vitro methylated lysine 943 by metyl21C. So here we wanted to further test the specificity of ARS1 methylation by metyl21C compared to the other metyl21 members, and to test if metyl21C can methylate the reported substrate, which are known to be methylated by other members of metyl21 family. So first, we tested whether the other metyl21 members can methylate ARS1. Importantly, you can see that none of the metyl21 family members can methylate ARS1 in vitro except metyl21C, as you can see here. Second, we tested if metyl21C methylates metyl21A substrate HSPA70 and the metyl21D substrate VCP, as had been reported in 2 different publications. Though in both cases, direct activity of metyl21C was not observed, not on HSP8 here and not on VCP. And as you can see here, metyl21A and metyl21D methylation on HSP8 and VCP used as a positive control. These data suggest that metyl21C does not directly regulate VCP or HSP8, and therefore the reported physiological functions of metyl21C in regulating these proteins may need reevaluation. So far, we showed that ARS1 is a substrate of metyl21C in vitro, so we would like to test if metyl21C interact and methylates ARS1 in cells. And this context co-immunoprecipitation assay from 293 T cell lines show that endogenous ARS1 interact with the flag metyl21C in cells. To ascertain whether metyl21C could methylate ARS1 in cells, GFP ARS1 was co-expressed with or without flag metyl21C in 293 cells, and the methylation status of lysine 943 was determined by mass spec. In the absence of metyl21C, expression of GFP ARS1 is not methylated at all in lysine 943. And in cells expressing flag metyl21C, the majority of GFP ARS1 is methylated at lysine 943, with prime methylation being the most abundant species. Importantly, using mass spec, we also found that endogenous ARS1 is mono, di, and trimethylated upon the expression of flag metyl21C in cells. Next, we raised a specific antibodies against mono, di, and trimethylated ARS1 epitopes, and we used these antibodies to test the methylation of ARS1 in cells. And consistent with the mass spec result, I detected metyl21C dependent methylation of ARS1 in 293 T cell lines expressing flag metyl21C. The methylation of ARS1 is almost certainly due to metyl21C, as depletion of flag metyl21C here using 2 independent CRISPR guide RNAs resulted in depletion of methylated ARS1. Based on these results, we conclude that metyl21C methylates ARS1 in a cellular context. So finally, I tested the methylation of ARS1 in cells expressing endogenous metyl21C. And as you can see here, the protein level of metyl21C is high in the skeletal muscles. And consistent with that, we see high level of methylation in this tissue, mouse tissue of the skeletal muscles. And IP using immunoprecipitation using the antibody of the di and trimethylation, we could detect ARS1. And Together, these results suggest that ARS1 is methylated at 943 in mouse skeletal muscles, a tissue type in which metyl-21C is expressed. So in summary, our study has identified ARS1 as a potential physiological substrate, and we speculate that this methylation might enhance the activity of ARS1 in cells. So here I just want to thank my PI, Orguzani, and my lab mates, and the organizers for letting me speak in this amazing symposium, and the scholarships from the TAT and the IDT that I got in my first year as a postdoc.
If you have a question from the crowd.
I have a small question.
Okay, sure.
So Mohamad, very nice work, a lot of biochemistry, and I was wondering if you have any idea what is going on in the regulation level, if you or someone else did ribosome profiling or something like that, to see how it actually affects translation in this tissue when you have these mutations in the activity.
Yeah, that's a great question. So the thing that we are now gonna do some kind of to understand the mechanism behind this methylation, how it affects the activity of ARS1, and as I say that might enhance the activity of ARS1 in cells and cells that expressing metyl-21C. So this is why we are gonna do the ribosomal profiling to address that and see if there is really effect on the translation level of in these kind of tissues.